everybody.
Good
evening. I like the enthusiasm. Tonight's speaker is Dr. Leo Ramos Ferreira. He was born in Coimbra, Portugal, but he comes to us today from far, far west of here. I don't know where west is right now. The University of California, San Francisco. Yeah. When I asked him, how did you get into science? He said that his first word as a child was not mama or dada, but sun, as in the star at the center of our solar system. While his interest started young, once he got to college and got into research, the excitement of being on the cutting edge got him hooked. His advice to delegates heading into the sciences is reasonably simple, yet potent. Do what you love, and don't be afraid to make a change in things that aren't working out. Outside of his scientific pursuits, as you will see tonight, as all of us will see tonight, he's interested in ballroom dancing, having been a part of the competitive team at Harvard, and traveling, where he particularly enjoyed visiting Asia. When asked, is the elusive Mothman real? He responded that he could be real, with any laboratory attempts to create the beast being possible with the right amount of NIH funding. And lastly, when faced with the age-old question, how much wood could a woodchuck chuck if a woodchuck would, he decided that the creature could throw a full stack of logs without the yard of wood. Without further ado, Dr. Leo Ramos Ferreira.
Good evening, everyone.
Good evening.
Thank you for that kind of introduction, and thank you in general to having me speak here today and give me the chance to experience science camp, or really any camp, for the first time in my life. So many smart people, and definitely now, I think the future is bright thanks to you guys. So tonight I'll attempt the impossible. I'll try to convince you that there's a lot of exciting biology besides neuroscience. So if you were to go to the doctor today, he would probably give you some sort of pill, a small molecule like penicillin, aspirin, and that would make your symptoms go away or just get slightly better. But it would really not get to the root cause of disease. Our body is made of cells, tissues, and organs. So you'd expect that if you have a disease of a tissue or an organ, maybe you want to regenerate, to repair that tissue. And so what would be one way, if you have a sick liver, what would be the easiest way to repair that sick liver, to get rid of that problem? Exactly. And so this was done first in 1954 in Boston, Harvard, where I was for 5 years. And so in this case, it was a kidney transplant. It was the first solid organ transplant that was successful. Before that, they all failed. And so the idea is, if you have kidney failure, you just give this person a new kidney. And so you could try to do this for different organs, but not so fast. Because we're all very different, especially when it comes to the immune system. So it turns out that if you want to fulfill this dream of regenerative medicine, where if you have a sick tissue or organ, you just replace that tissue or organ, maybe coming from someone else who has a lot of excess of a tissue or organ, is not so easy, because there is this barrier to really accomplish just putting all these healthy tissues in you to repair that problem. And so that was first unveiled also around this time in the last century. And the idea here is very simple, was that researchers noticed that if you have two strains of inbred mice that are different, and if you transplant tissues between these different mice with the same strain, they are accepted, no problem. You can take a skin patch from one mouse to the next mouse, and nothing bad happens. This is the longevity of the graft. It's infinite, let's say, at least for the amount of time that they looked at it. But if you were to take skin from this yellow mouse strain and put in the blue mouse strain, it gets rejected. And even worse, if you do it again to the same blue mouse, it gets rejected even faster. And so what this suggested was that there must be something that tells the blue mouse that his yellow piece of skin does not belong to him. And so these three researchers, Vanasarov and Dosey and Snell, were able to, using very experiments find out what that region in your DNA was. So what they did was, every time they just crossed, and they crossed a yellow mouse with a blue mouse, so you get like an intermediate mouse. They would take tissue from intermediate mouse into the yellow mouse. It gets rejected. Okay, now we cross that progeny again into the yellow mouse. And you keep repeating these experiments until the yellow mouse and this yellow with a bit of blue mouse differ by just one locus in the genome. And that is the MHC locus, major histocompatibility complex. And so this locus goes for an array of genes that come in many, many flavors. Like if you think Baskin-Robbins has many flavors, this is way more than that. HLA-A has 5,000 different alleles, that's why we call gene flavors. HLA-B has 6,000, yes, I'm for a word, and so on and so forth. And so what this means is that all of us in this room, it would be exceedingly rare to have all 12 alleles, so have 3 from HLA class 1, mom and dad, so that's 6, and then 3 from HLA class 2, so that's 12, that they all be an exact match. And so what that means is that no one in this room would be able, just by chance, to donate an organ to someone else in this room without it being rejected right away. And so that creates a big problem, because if you have this mechanism where you have your immune system cells, your immune cells, and they know that there's something wrong. So the MHC molecule has this binding group. And so it will present pieces of proteins from inside the cell. We call them epitopes. Their peptides are immunogenic, recognized by the immune system. And so there's a peptide group. And so then comes the T cell with a T cell receptor. And the T cell receptor will recognize this peptide MHC complex, and will scan it and tell you if it's self or non-self. Because the way it works is that when we're born, we have a set of T cells that they can kind of predict the future. Like if we encounter a virus later in life, we already have a T cell recognize it really well, just by chance. Because it always strikes the balance between being not too weak, such that a T cell is non-functional, and not too strong, such that it would attack your own tissues. And so when you have the right amount, it's a self peptide. When there's something wrong about it, it's a non-self peptide. And of course, T cells, there's come in 2 flavors. There's CD4 T cells, CD8 T cells. CD8 T cells are the killer T cells. They see MHC class 1. So there's this nice hot dog over here. And every cell in the nucleus in our body has it. So what would be a cell that doesn't have MHC in our body? Red blood cells. Red blood cells are just like biconcave discs. They don't have MHC. They don't even have a nucleus. So there's nothing really to talk to them with. But then you also have helper T cells. So the helper T cells are essential to mount a strong immune response. They're essential for memory. That's how vaccines work, for example. You expose yourself to attenuate to a weak version of a virus, and then your body builds memory. So when you encounter the real deal, your body knows what to do and eliminates that virus right away. And so that's the MHC class 2 that will talk to the CD4 T cells. And so those CD4 T cells are lazy. They don't really go to the actually infected cell. Instead, they talk with these professional antigen-presenting cells, starting as themselves, it seems. And so these APCs are macrophages, dendritic cells that crawl around all your tissues, and they're constantly picking up stuff from the environment. And so that's how they pick up, they can incorporate that inside them, and then present it once again on the surface, such that now the CD4 T cells, the helper T cells, can see it. And so that's immunology in 3 minutes. So you have this, and so now this is a bit more detail. So if you look inside the cell, like proteins don't live forever. They're not like diamonds. They are inside the cell, and then the proteasome will degrade those proteins into peptides. And so those peptides can be picked up by this protein tag that can then load them into MHC molecules together with the accessory chain, beta-2 microglobulin. I want you to remember this gene, beta-2M. It's going to come up later. And so once you have that whole complex, it goes to the surface of the cell, and there's a T cell that can now scan and see self versus non-self. And so what that means is that this whole maybe original idea that can just like pass organs around and cure everybody is, not so fast, because there's immune rejection. And so this is a really big problem, because actually 10,000 people die every year because they cannot find a matched organ to their disease. So they just die in the waiting list. Or sometimes people compromise and they give an organ that is not exactly matched. And it might not be rejected first week, but it will be rejected over the course of years. And so those people, they lose their organ and they go back in the waiting list. And so that's the United States alone, the 11,000 people. And so what would be, and so on top of that, I guess before that, so many people waiting for a kidney. And then I should also say that if it's not exactly matched, there is a way to prolong that acceptance. And those are called immunosuppressive drugs. These are very blunt drugs. That what they do is that they basically shut down T cells. They attack the T cell cycle, their capacity for memory. And so what these do is that, what it says that these people now, yes, they keep their organs for longer, but if they get a common cold or if they get like a blood cancer, they're extremely susceptible. So even if they don't die from the organ failure, they could die from a viral infection or from cancer. And so what would be a solution to this kind of problem, right? Because we can't just, if you look at where these transplanted organs come from, it's a very wide imbalance. Because you have poor countries being on the top list of organ donors, and you have the developed countries being on the top of the recipients of these organs. And so you even can imagine like a black market, these are all real images, where you can imagine people that, they have to feed their families, and they're really in a desperate situation. And they literally have to put their parts of their own body for sale because of this problem at a global scale. And so, okay, what would be a solution for this shortage of organs, of compatible organs? Like, what can we do to just try to solve this? Yes?
Possibly you could create like synthetic organs.
Okay, synthetic organs, yes.
Suppress immunogenic responses.
Sorry?
Suppress immunogenic responses.
Yeah, okay. Yes.
And the existing organs.
Sorry?
And the existing organs.
Yeah, so maybe if you can really get down to the mechanism, can actually go inside that person, so giving them a new organ, just repair the endogenous, okay?
Modify the person who's getting the organ's immune system, or modify the cells of the donor organ.
Yeah. Okay. Yes?
3D print organs.
Sorry?
3D printing organs.
Okay, so we have synthetic organs, recruited organs, so we modify the immune response. Or, okay. Anyone else? The last? Yes.
Prosthetic replacements?
Sorry?
Something
Oh, yes, so maybe you like for the heart, maybe you can have just a pacemaker, like a mechanic thing. Okay, one here.
Use part of the original organ that was broken as sort of a template or use part of it or try to repair it as it works.
Okay, so maybe you can summarize it as a seed of like as a template, as a scaffold to make a new organ. Okay, here.
Change immune cells to make them recognize the new organ itself.
Oh, I like it. That's what I'm working on. Yeah. Wow. You guys are too good. Okay, yeah.
We could modify the laws, and instead of opting in to donate, you could only opt out of donating. But if you opt out of donating, you can never get an organ, and you can't participate in universal health care.
So actually at UCSF, every year we get a fellow from Japan because in Japanese culture people don't really donate organs, and so they come to UCSF for a fellowship because UCSF has more volume of organ transplants than the whole country of Japan almost. So that's how low their rate is. Okay, so there's many ideas here obviously, so I'll go through some of them. But so the first one I guess would be, what if you can just make the organs? We need to take the organs from someone who just made them. And how you make them? From embryonic stem cells. Who knows what stem cells are? Just raise your hand. Okay, so what are the two properties that a stem cell has to obey to be called a stem cell? Who wants to tell me one of those? Yes.
They can develop into any type of cell.
Okay, yeah. What's the second one?
They must not be specific cells. They must not be specific
Yes and no, but that's not like a formal rule. Because you'll see there will be embryonic stem cells and there's adult stem cells. Actually, go back to your report. So if you have an embryonic stem cell, as the slide already tells, it can give rise to all three layers in your body. But if you have a blood stem cell, it can only give rise to blood. and yet we still call it a stem cell. Actually, the first stem cell to be discovered ever was a blood stem cell. No, big things happen when we work with blood. Okay, what's a second rule for a stem cell? We call it stem cell.
They have to be able to express every single gene, or at least they have to be able to express every gene.
At some point, a differentiation patterning, not as a single cell entity, correct?
Yeah, just they have to have a potential.
Okay, yeah, so pluripotent or multipotent, yeah, okay. So that's still more of the same. I want the second one. Yeah, now you can go.
We have to involve science, technology, engineering, and math.
Is it an FLA? 5-letter acronym? Okay, so the second, okay, you go.
They can create more stem cells.
Oh, yeah, okay, so how would you rephrase that? A stem cell can create, okay, yeah.
Oh, isn't they can regenerate?
Yeah, so they divide indefinitely. They're immortal. Stem cells live forever. So stem cells can divide indefinitely, and they can give rise to all cell types in the body. If it's a pluripotent stem cell, has pluriparts, or to specific tissue, like a blood stem cell, all the blood cells. A muscle stem cell, all the muscle cells. There's only one cell, that's why it's a formal definition. So the muscle stem cell, which you activate every time you work out, it's a stem cell because it defines indefinitely, and it gives rise to all the cell types in its tissue, which turns out to be only one cell type. So it's like a one-to-one. And yet it's still a stem cell, but it's one or more cell type. Okay? So that's what a stem cell is. And so, okay, so one way, okay, so what would be one way to get embryonic stem cells? Like how do I get them? You go.
Take the skin from the person and then you transform it into a embryonic stem cell?
Okay, how about something more direct? But I'll go back to you.
Is there like a tube, like from like the baby, they have embryonic stem cells, or can we just use IPS?
Yeah, so I guess the embryonic stem cells, by definition, are from the embryo, from the inner cell mass. So you have a blastocyst that's being implanted, and there is where the embryonic stem cells are. So they have to take out an embryo and isolate the stem cells. But you can imagine that like ethical implications. It's not, it's not cheap. You can't really scale it up. So instead, and I think you already know everything, right? But so there's this very bright guy, Shinya Yamanaka, in Japan. He got the Nobel Prize in 2012. And what did he do? He told his postdoc, which is what I am right now, these genes are expressed in stem cells. And they seem to be specifically upregulated, so more highly expressed in stem cells versus other cell types in the body. And previous experiments had shown that if you fuse a stem cell to a differentiated cell, so we have a single cell but two nuclei, what do you think happens? Does the differentiated cell become a stem cell, or does the stem cell become differentiated? Like, what do you think happened in the experiment? Actually, no. So the differentiated nucleus became a stem cell. And so what that proves is that whatever it is that stem cells have, it's a dominant factor or factors. And so what this guy did was he took 24 transcription factors. These are proteins that bind DNA and change the expression of genes. They change how much mRNA is being made from those genes. And he made viruses, viral particles of all those 24 genes, and he infected fibroblasts, so skin cells, from a mouse first. And what he found was that if you take those 24 and put in a skin cell, you can revert the clock. And now the skin stem cell, the skin cells or adult cell, became an embryonic-like stem cell, which equaling in this pluripotent stem cell, or iPS cell. But then 24 is a lot of virus to make, and so you kept going down and down and down like a puzzle, and narrowed down to 4 factors. So it turns out that if you have 4 factors, OX4, SOX2, KLF14, and c-MYC, if you have those 4 transcription factors, that is enough to convince any adult cell tested to date to revert to an embryonic-like state. And so, hence, that's the birth of man-made stem cells. So I no longer need to go refer to embryos to get pluripotent stem cells. I can just make them myself in the laboratory by taking nowadays a blood sample, which is more accessible. Like actually my professor from graduate school, he still has like a scar from a skin punch that they did to him to get skin cells. But with blood, no scar. So again, blood is just awesome. Okay, and so then you can imagine you can make these, and so if you have a tissue that is failing, you can take that patient, make stem cells from that person, differentiate them with the right recipe in the tissue that is lacking, and now whatever disease that person has will go away. And so this is happening right now, not just by PSO, but also other those stem cells in general. You know, we've heard about hundreds of millions of dollars. Like when it's talking about healthcare, we talk about billions of dollars. In this case, the market is $32 billion. $32 billion is the market for regenerative medicine. So this is all good and great, but what is a drawback? Or there's probably many drawbacks, obviously, but what is an immediate drawback of using your own cells to cure your disease? Yes?
It takes time to grow.
Okay, yeah, so it's a laborious process. Like you have to take the skin cell, you have to differentiate iPS cells, so you have to make the stem cell colonies, pick the right one, use the recipe, differentiate the one. Okay, what's another drawback?
Did it just happen again?
And why would it happen again?
Because it's the same exact thing.
And okay, no, it's okay. That's not about the essence. Okay. Okay, all the way in the back. Okay, so maybe it feels like some sort of dominant aspect of the disease. If you just have more of the same, it make it worse. Okay. So what about the essence? Okay, so what is it? Like, what is the essence?
Like, if it's the same cells that have the same, most of the same genomic code, then it's susceptible to the same disease as some person.
Exactly. So if this disease is caused by a genetic mutation, then if you're just making more of the same cells with the same genetic mutation, that's not going to solve anything. Well, at least not in the long term. The easiest, clearest example would be sickle cell disease. Sickle cell disease was the first molecular disease because that's when we really knew that a change in a single base pair of DNA will then cause a base in a single amino acid. It's the cause of the disease. So if this person has sickle cell anemia, such that the red blood cells have that sickle shape, they'll transport oxygen very well. And if you were to just, take stem cells, so make iPS cells from this person, differentiate into iPS cells, then red blood cells, and put them again in, the red blood cells will be just as defective as one you just took out. This is like a very extreme, obvious example. We can imagine more, like subtle examples where if you have the disease gene there, then it's going to be more of the same. Okay, so I have a mutation. So what can I do to address this then?
Use CRISPR transmutation.
Okay. So CRISPR. So that's right. I first learned about CRISPR in 2012. I was in graduate school, and my program has this weird rule that you've got to present a paper, an article in Journal Club, where you cannot use PowerPoint. You have to draw every slide yourself. But if you draw the slide, you think about it 3 times. Instead of like flash PowerPoint after PowerPoint, you think about it first. You digest the information. Number 2, it has to be completely unrelated to your project. And so I was in an immunology lab. And so, so work with human cells and mouse cells, so I picked a bacterial paper. So this paper, they show that some bacteria have this system, this CRISPR system, where they have this sequence that they keep from viruses that infect them, and they have this nuclease, Cas9, that can cleave double-stranded DNA with 2 domains, which is the HNH and RUFC, and so they do so when that sequence of DNA is recognized by the guide RNA, which is composed by a CRISPR RNA and a trace RNA. Anyways, a biochemistry paper in bacteria. So I was like, okay, I'll read this paper. Sounds interesting. I'll present it. I'll never hear about this ever again in my life.
So you presented the Zubda paper at Harvard?
Yes. This was before the patent fight. This was back when we were all friends. And so this was in like October, November 2012. Come January 2013, 3 papers back to back show that we can repurpose this system to edit human genes in living human cells. And so we go from this obscure bacterial system into, okay, we can now rewrite the genome and cure all diseases. And actually, it started, CRISPR was discovered in a specific type of food. Can anyone guess what type of food it was? Not chips. Yogurt. Yes. because bacteriophages are bad for the yogurt, big yogurt. Big yogurt does not like bacteria. I might be sponsored by big yogurt. So bacteriophages will like kill off your bacterial cultures that make that delicious yogurt. And so they found that some bacteria somehow will resistant to this lytic cycle, right? So the bacteriophage will go, there's a lysogenic cycle, there's a lytic cycle, and the lytic cycle is when the virus goes, invades the cells, makes as many of itself as it can, and then the whole thing just explodes, and they go colonize other bacteria. It's a bit like us. We're on Earth, and we go to Mars, and we just keep going. It's a bit akin to that. So what does CRISPR stand for? That's right. And that's because he was first identified by a Japanese group. And so they just sequenced bacteria, like they did sequencing was a hot thing back then. And so they just found that several bacteria these like weird repeats. And then there was a Dutch group, I mean, leave it to the Dutch, to say, well, I know we're going to call this guy Leo. Well, what did he think? Well, I'm going to call this cluster regular interspace short palindromic repeats. They're short, they're palindromic, so if you swap them this way, they still read the same way. They're repeated, they're in clusters in the genome. So there you go, CRISPR. Nothing to do with fridges or freezers. And so then came, again in yogurts, they found that then if you actually integrate this CRISPR system in bacteria from different ones that were previously shown to be resistant to a virus, it becomes resistant to this new bacteria. So this property is transitive. So you can take this system and really make that this new bacteria be resistant to it. So then what we now understand is that, again, it's like kind of adaptive immune. I can't even say this, but it's that bacteria like an adaptive immune system, in that a virus will infect the bacteria and will have some DNA that gets incorporated by still poorly understood mechanisms in its CRISPR locus, and then in the future infection by the bacteriophage, the bacteria knows exactly what to do. Have the Cas9, have the guide RNA to that specific sequence, they'll just snip it out, and it can now resist infection by this bacteriophage. And so that's the 5-part where basically you have on the West Coast Jennifer Doudna and Emmanuel Japanese in Europe actually. Then the East Coast you've got George Hirsch and Fong Chong, both of them published January 2013 and patented use of CRISPR-Cas9 in human cells. They're fighting to this day. They thought it was something, but then they just renewed a new lawsuit this year. So what this means is that now you have, it's like you have a Word document. You can think about it the same way for your genome. So if you've got like a disease gene, you can replace it with a correct gene. And so now this gives us an idea like we can now have medicine with molecular precision. So now we can go and like fix. But you know, science is slow, science is hard. And so we thought that we'd start with something simple to prove that we can use CRISPR-Cas9 to bring this precision medicine to life. And so back then, if you look at a timeline, you see that CRISPR had been used in different cell lines, but never actually in primary cells. And so, what is the most accessible tissue in the human body?
Blood.
Is it, okay, who wins, the skin gang or the blood gang? Okay, so who says skin? Oh, wow, okay, who says blood? Okay, pretty tight. So I myself vote on blood.
Yeah.
So if I take a piece of skin, it's going to hurt. But if I take some blood from you, yeah, actually, you can't take my own blood because I'm cancer. But that's me, I'm the exception. So blood is the one tissue that it's not in one single place, you know, it's all inside your body, and you can take it out at any location, of course, from the veins, and you can actually develop very good protocols to engineer those cells and put them back in. And so I thought, well, can we use CRISPR-Cas9 in human blood cells? And so, again, because science is hard, we thought, okay, let's start with an easy problem. So we focus on AIDS. So AIDS is a global pandemic that affects millions of people worldwide, and we know how it's caused. It's caused by the cells based on a C4 receptor and this other receptor, CCR5. And so you see that over time, as you get the infection, the number of CD4 T cells will like sharply decrease. And remember, from my 3-minute immunology course, CD4 T cells are the helper cells that are required to have a robust immune response. So once again, a bit like an immune suppression by drugs, these people, if they have an infection, if they have a blood cancer, they can't really fight it off. And so that's why, so they will die. They have a very high degree of morbidity and mortality because of these secondary infections. And nowadays, it's become more of a chronic disease, and that's because we really carpet bombed it. So we studied HIV for decades now, the HIV virus, so we know a lot about its cell cycle. So people will take up to 5 different drugs that will block integration of the virus, that will block production of viral proteins, that will block the entry of the virus in the cell. And even then, because the viral genome of HIV is very small, and the smaller the genome, the faster it mutates. So it's an arms race. It will be doctors with drugs versus HIV, and HIV so far almost always wins. And so we thought, well, this is not sustainable, so can we use genetics to find a solution for this terrible disease? And so luckily for us humans, some people actually won the genetic lottery. Because even if they're infected with HIV, they never get AIDS. And that's because they have a mutation in the CCR5 receptor that is required for HIV to enter CD4 T cells. So if you have a homozygous mutation in CCR5 gene, which happens in 0.01% of Nordic Europeans, you never get AIDS. You'll carry the virus, but you'll never get AIDS. And so we thought, well, we know that this could work upon transfer because the Berlin patient, which was actually American, was studying Berlin at the time, he got cured from HIV and leukemia at the same time with a bone marrow transplant from a compatible donor that happened, well, the doctor showed it that way actually, let's be honest. They have a homozygous delta 32 CCR5 mutation. And so, if some people win a genetic lottery, and this can be conferred upon transfer of bone marrow, which active ingredient is the hematopoietic stem cell, HSCs, or blood stem cells, can we then do this for everyone? Can everyone be a winner? And so we went ahead, and so we thought, okay, so we take the CCR5 gene and we do a gene ectomy. We're going to excise that portion of the gene in human blood cells, such that now you'll have blood cells that have this mutation. So from this long list of cells, which one do you think we targeted?
T cells.
Why T cells?
Because it's what the HIV virus attacks.
Okay, and there are indeed clinical trials with CCR5 deletion in T cells. What other cells would we have tried? Or maybe we tried that cell.
So
Okay.
it kind of comes down to treat all generic cells.
So HSCs, and that's because the HSC gives rise to all blood cells. So if you want a longer-term cure, you should modify the HSCs because, reminder, they divide forever and they give rise to all cell types in the blood tissue. And so you can imagine if you have HSCs that don't have this mutation, they will, 2 things. They'll replenish, they'll make a whole new blood system for yourself, number 1. Number 2, this new blood system will have T cells that will recognize HIV-infected T cells without they themselves being infected with HIV because they're deaf to it. And so that's the principle here. So basically give you a new blood that is resistant to HIV. And so we tested that would be possible, and so who knows what this is? Like, what is this technique? Shall I show you? Look, how does it work? Like, what is this? Okay, what's this band? This is DNA. What about this band in relation to this band? What's the difference?
It's size.
Okay, which one is bigger? The upper one or the lower one? The upper one, right? So if you have like an obstacle course, and you have this agarose mesh, and like the bigger, so DNA can make it linear very easily, so, you know, like the bigger the DNA band, the further the less far it can get if you run for a given amount of time. So this DNA band is lower than that one, so that means it's shorter than that one. And so why does this matter? Because actually, these 2 bands were produced by the same exact PCR reaction. We know PCR, right? Polymerase chain reaction. So you can amplify energies of DNA that you want, with flanking primers, so welcome CCR5 gene, and so that's the exon there. And so if you amplify primers binding there and there for a specific region, and so this region will come out with the different size. Why? Because in some cells, my CRISPR experiment was successful, and so now that region is literally shorter, right? Because when you cut the DNA at that specific location, the cell had two choices. You could repair DNA with HDR, homology-directed repair, where it will use a neighboring sequence that looks just like that one from a sister chromatid, and just, give it back to normal. Or, way more often, it will use non-homologous end joining, NHEJ. And so this is, this system of repair is not very precise. So every time you join the end of the DNA back together, because otherwise the cell will die quickly, maybe it will miss a couple base pairs. So what that means is that if you imagine a protein with amino acids and codons at the DNA level, if you just leave out one letter, the whole thing shifts. And so because it shifts, it's all screwed up now. You just read something else different. So that's how we functionally do a knockout using CRISPR. And so what this means is that we're successfully editing both copies, right? When we have mom and dad, for example, here, you have one copy was edited, the other one was not. That can happen, right? You have two copies. But if we're efficient enough, we can get both copies edited, no trace of the wild-type copy. And so we can edit ourselves. Okay, so I have this petri dish with these HSCs that don't have the CCR5 gene being expressed. Now what do I do with them to test them to see if they would work? Like, what's the next step?
Sample infected with HIV.
Yes, okay. What else?
So it's like a T-cell RFP. Take these embryonic stem cells and actually develop them into the cells before producing the parents.
Okay, so see if they still get infected with HIV, see if they can give rise to the cell types as they should. Okay, what else? Okay, you'll go again.
They can forget ethics and just put the cells in.
Into what?
Let's just not be ethical and just put the cells in and see what happens.
Into people, into yourself?
Yeah, into the person who's cells.
Or not?
You are not.
Or yourself.
Yes, if I was born to get HIV, yes.
Because these are your own cells, right? Yeah, okay. You had something, right?
Did they start off with testing it in mice to see if they develop AIDS?
So mice, HIV does not infect mouse cells. So the HIV infects human cells. There's SIV, which infects primate cells. There's no HIV for mouse cells. But maybe we can bridge these two worlds, it's, maybe we can have a mouse that somehow is human blood. So that's the human mouse. So this mice have a genetic mutation. He is doing fine. These mice have a genetic mutation that deprives them from the development of B cells, T cells, and natural killer cells. So all lymphocytes. It's a deletion of the IL-2 receptor common gamma chain. What that means is that there is cytokines, these growth factors, that are required for T cells and B cells to grow. If you don't have those, you don't have T cells, you don't have B cells. There's actually the bubble boy and such mutation like X-skid. So that's what the bubble boy is. It does not have lymphocytes. And you can make the same thing happen in mice. So then you have a mouse that doesn't have its own lymphocytes. So we're going to help him out. And we're going to give him human lymphocytes. So from human fetal tissue, you can take 3 different tissues. And so who knows what BLT stands for? Not anymore. BLT stands for bone marrow, liver, and heart. So we get fetal samples, and we get human fetal liver and human fetal thymus. So thymus is a little organ up here. That's where our T cells go to school. Actually T cells are called T cells because they are educating the thymus. and B cell in the bursa of Fabricius. Someone really liked chicken back in the day, and they worked with birds only. They found this organ in birds called bursa of Fabricius. His name was Fabricius. And so he found these lymphocytes called B cells. So we have B cells and T cells. So either way, fetal liver, unlike in adults, is a site of production of bone marrow. So for us, it's in the bone marrow is where we produce the lymphocytes. But in the fetus, in the human fetus, in the mouse fetus, is the fetal liver that makes those cells. So you take the liver, take a little piece of liver, a little piece of thymus, and then from the liver actually can go and use what's called FACS, fluorescence-assisted cell sorting, to purify hematopoietic stem cells from the fetus. And so when you put those three things together, and you chop it up and you went to different pieces, you can make a batch of these mice. So this again are mice that have their own lymphocytes, but they have human lymphocytes now. And so what we can do this for is we can now have, maybe we have wild type HSCs, or we have our own edited HSCs, and that's what happens. And to our pleasure, if you see, they can differentiate into the main branches of the immune system. So if you think letter acronyms are confusing, you don't know immunologists. We don't stop at letters, we actually use numbers. So CD45 is a marker found in all blood cells. And what for us, human CD45 is distinct enough from mouse CD45 that I can raise an antibody that will bind to one but not the other. So that's the first step in this data, is that in this panel where each dot is a cell. We did single cell way before it was full. And so this dot here, for example, and all this population is like a seismic clock. Like when you have too many dots, you can see how many more just like this curves. And then the more concentrated, the more cells you have in there. So you have human cells in the mouse. That's good. And from those human cells, you gate on that, we call it. You just, net the cells, next plot. And you see that some of them are CD19 positive. Again, meaningless number. It just means they're B cells. And CD11B, again, meaningless number, means that they're macrophage and dendritic cells. And then the ones that are neither that nor that, we then take those and look. They are T cells, they have CD3, and not more than that. They actually CD4 T cells. So all this to show you that if you take these modified HSCs and put them in this BLT mouse, they still make all blood cell types. So now we're ready to go. And more importantly, so I have this, what piece of it is missing to really convince you that I am able to generate CCR5 negative HIV resistant blood inside a mouse? What's missing here? Okay. Okay, right. So maybe all the CCR5 edited cells died, and all I have these wild type cells left. But fortunately not. When you do the genomic DNA isolation from these cells, you still see that a portion of the cells that they remain in the mutation. So again, from this mouse, so in humans, there's see the summary. So you have your donor. He gives you mobilized peripheral blood, so this M stands for. And this is important because usually, if you want to sign up to be a bone marrow donor, and you do have to give bone marrow, how do you extract the bone marrow from you?
You can just take, you can just take it out of a large marrowless bone. Or I think what you're probably going to talk about is that nowadays they can have you release those stem cells out into your blood and take it right out of your blood.
Exactly. So you can give drugs to people that their HSCs will sniff them and they'll come right to your peripheral blood. And so you just do a regular blood draw, and then you can actually purify HSCs from that regular blood draw. Very good. So you take those cells, you purify them, then you reach for CD34, another meaningless number that means the HSCs, and then you nucleophect them. So you have the cells, you have at the time the Cas9 DNA, the RNA DNA in a cuvette, and you electroporate it. So the membrane will open holes, and the DNAs go inside. And so, we like to see to believe. So we label the Cas9 with a 2-AGFP. So every cell that's got Cas9 also got GFP. So we can sort them. So there's a sort there. And so the idea there is now, once you have your purified cells, you can now, as I showed, they have high efficiency. They have intact function. So in the mouse, they can make the different blood cell types. Actually, also in vitro, you can have this quality-forming assay where you can convince your HSCs to become different kinds of blood cells in vitro, but in vivo veritas. In vivo is always more powerful evidence. And also importantly, no off-targets. So you can imagine that CRISPR, just like any gene editing tool, it cut here for sure, I show you that, but maybe it cuts in 1,000 other places. So we did very careful 3,000x sequence of genome of the cells to show that there were no off-target effects. Not with all guys. With one of the guys that was true. With other guys that was not true. So that means that going forward to the clinic, what I think has to happen is that you will need to sequence the whole genome of the patient receiving these treatments. Because none of us has a reference genome. And so just because one person does not have an off-target site on her or his genome, does not mean that the next person does not have it. So you need to sequence the whole genome, I believe, and then map out what could be the off-target sites. And so in our particular cells, from our different donors, there was not a problem. There's no unique off-target that just popped out. So that's the summary of that part. And so the idea again is that you can hopefully help treating this disease by having these modified cells. So this is in blood, which is a very available organ. That's not going to be as easy for other organs. Right? If you're like in the liver or in other organs, like are you going to inject the Cas9 into you? And the guide RNA, are you going to get to the right place? That's not easy. So maybe instead, so it's much easier to deliver RNA than, giant genes. So maybe you can just in the future make humans that already have Cas9, and then the drug will just be the guide RNA. So if you have any problem, so you think, no, that's not ethical, we can just modify babies. Well, too late. Too late. I did not do it. So our publication came out in 2014, to modify CCR5 in human blood cells. This guy, 4 years later, showed, Lulu and Nana. Hello, say hello to the world. These two human babies also have mutated CCR5 induced by CRISPR-Cas9 at the embryonic level. So when you have the in vitro fertilization, you can literally inject with a tiny needle, the Cas9 and the guide RNA into the fertilized human eggs. And you can use the IVF procedure, and then you can implant them into the mom. And so she had these two babies that when you sequence their DNA, they have mutation CCR5. And so I'm not saying I'm supporting it, I'm just saying, I think as the last speaker said, it is in our blood to explore and push the limits and maybe make things worse for us in the far future. But as of now, this is happening. There was an article in Nature with a Russian scientist saying, yeah, I think I'm crazy enough to do it.
Oh, no.
And so this is going to be, like the moon race is going to land first in the moon. It's going to be the same thing for CRISPR editing in people. And so this is a crazy time that we have right now. In 2013, Human Cell Lines was next door to me, Broad Institute at MIT. In 2013, also next door to me at MIT in mouse models. So back then, mouse models very long process. You have to do all this almost recombination. Now this paper, they could do 4 gene knockouts in mouse embryos, just like that. And in 2014, we did it in human blood cells. 2015, 2017, we did it in human embryos, but they actually didn't bring them all the way to becoming people. And then in 2018 was the first clinical trial with CRISPR. CRISPR Therapeutics, actually founded by one of my PhD advisors. And also, what I just showed you, that actually in human babies now. And so that's why I can go talk about this, can you now take this beyond blood? And so one example that I thought was really cool, so I share with you quickly, is that recently it's been proven that not just blood stem cells, but also skin stem cells formally obey the definition of adult stem cell, because they divide forever, and one single cell can give rise to a whole tissue. So I had a patient with this terrible disease where the skin literally boils away, hence his name in Latin, bullosa. And so you guys are not sensitive, I know that, and so that's the patient. And so they took whatever patches of normal skin were left. This was in Italy, no, in Germany. They sent them to Italy, and they then isolated skin stem cells, modified them with that vector that contains a wild type copy of LAN3. This is good old gene therapy, so no CRISPR-Cas9 or anything. Just put in the correct gene and hope it doesn't land an oncogene. Grow those cells as these patches of skin, and then ship them to Germany and transplant them to the patient. And the patient is actually doing great, as far as we know. So once again, you can make whole shreds of skin And then when they sequenced the skin, they saw, that's what this animation is coloring, is that really the whole skin came from only 2 adult stem cells when they sequenced the DNA. Like you can trace it all the way back to a single cell derived clone of skin stem cells. So what that suggests then is that we can use the stem cells and modify them for therapy. And so can we then, expand this for the embryonic stem cells, for embryonic stem cells. So I told you remember about beta 2M because you can imagine if you knock out beta 2M, then this complex never gets assembled, and the T cell never gets to see anything. So these are you making invisible cells. The immune system, at least the T cell part, cannot see them. On top of that, we can also introduce genes that induce what we call immune tolerance. So immune suppression is when you shut down the whole immune system. Immune tolerance is when the suppression is antigen specific. It will be specific, suppress the response to a specific epitope. It's that one piece of that one protein that the immune system leaves alone now. And so what is the craziest case of immune tolerance naturally occurring in humans? Right, the case where somehow the human immune system tolerates non-self for a long time, for many months.
Pregnancy.
Yes. We're all here today thanks to successful pregnancy. And for immunologists, it's really a mystery because the fetus is 50% foreign. So by the basic mode of transportation I showed you a few slides before, it should be rejected. It's got foreign antigens, and it's really in close contact with the maternal tissue. So actually the fetal cells will go, they'll invade the maternal vessels and open them up, so they increase the blood flow. The maternal immune cells extensively contact the fetal tissue, and yet it's like nothing has happened. So there are molecules that we don't fully understand yet that are expressed by the fetal part of the placenta that shut down the maternal immune system locally. If you give a vaccine to a pregnant woman, it still works. If you take a skin patch from her husband to her, it still gets rejected. And so this is the ideal case. If you have an organ transplant, you want the response to be shut down only to that liver, only that location. You want the rest of the immune system to be completely untouched, so you're a completely healthy person. And so that's what we're working towards. So we use this assay, again, in our favorites, immunodeficient mice, called a teratoma assay. Who knows what a teratoma is? What is a teratoma? Yes. Yeah, this is pretty good. Anyone wants to add anything? Or? Yes. So, yes, you want to add something?
Is that also like one of the things that are involved in chimeras? Like unpotential for chimera?
We'll get there. So, okay, so teratoma, as you said, is a tumor. It's a developmental tumor. It can form all 3 layers inside it. So, if you're lazy like I am, you're going to see, okay, can I protect most cell types from immune rejection? If you do a teratoma assay, you put these stem cells in, they'll make different cell types, and if all these cell types are protected, then you kind of prove that you blunted the immune response against different types of tissues. You can go later on in a more careful experiment, but I'll just show you this one quickly today. So again, you have your different embryonic stem cells that were modified. So you have your traditional knockouts, so no MHC, but you also have these ones with different tolerogenic molecules. So see if on top of not having MHC, if you can make them even better at being protected from the immune system. So I'll just show you the first part, which is if you delete beta 2M, hence surface MHC class 1 expression. So the T cells have nothing to grab onto it. And so if you look at the T cells in this teratomas inside the mice, you see a very drastic difference. If you look at the normal, the tumor gets rejected. How do you tell? Not from the size, but from the consistency. This tumor's consistency is cystic. It's filled with fluid. It's rejected. Whereas this one, the beta 2M double knockout, it's more like, it's more like meat, more like a steak. So if you look at that, you now have this evidence, this in vivo evidence, in vivo veritas, the evidence that these tissues are now protected from T cell attack. That's what we're exploring now. And so the idea is that, like, we have a universal blood donor, so type O is negative. Can we have the same thing but for stem cells? Can we have universal stem cells where we can, give them to anyone? We can make tissues, and so I think at the beginning what will happen is that we'll do chimeras, and the chimera will be, we now know so much about development that we can inhibit the development of any organ that we want. For example, PDX1 is a transcription factor that's required to develop a pancreas. So what is an animal whose organs are roughly the same size as ours? A pig. So you can have a pig embryo, delete PDX1, so this embryo is not viable because you never have a pancreas. But what if I add some human universal stem cells in? Now this pig will be born with a human pancreas, and you can then harvest that pancreas for transplant to a person. Any person, because they're universal stem cells. And so just like we have farms of pigs now, we can have farms of these pigs that will now have human organs. So I'm not saying it's a pleasant idea, but I think it will be, like many things in science, but it will be probably the first step. A much better step is just go on Amazon Prime. And just say, I have a heart, please. So someone 3D printed a heart, you know, I have a scaffold. I think your idea is all excellent, right? Like a scaffold from original heart. You see it with stem cells. You then differentiate to beating heart muscle cells. And then you make the right shape, right size that you want. And you just click, 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 click. And they make it for you and they ship it to your home. So that would be the ideal, right? Is to just make these organs live. And so this is what's being thought right now. Certainly, art has been 3D printed. It's not a functional art. So that's why I think animal step will still be required with large animals, but that'll be the future. And so, you know, this has gone to industry, the license go for patents, and so the idea is again, you know, any cell, any disease, you know, it's all about selling the idea, but honestly though, like if you can, I think the more we do this, the cheaper it will become per person, and the more feasible and more people become interested in this. And so I think we should just so dramatically change the way we do medicine. And so this will lead to the last 3 slides of my talk, which is what I'm working on as of last week before coming to campaign. I lost my signal, which is, can we re-educate the immune system? So maybe I don't need to change the graphs. Can I put myself in the shoes of the immune system? And so can I change the opinion of the immune system? Okay, maybe this organ is not that bad for me. And so luckily for us, there's a very special cell type in the immune system. It's like the general of the immune system. And that's the regulatory T cell, shown here in purple. This schematic is not for understanding purposes. It's just to show you that the T ray touches, I believe, every immune cell that is out there. And so what it does is that it will suppress specific immune responses. And so if we can coax these versatile cells into becoming living drugs, we can now go to organ transplant or even to autoimmune disease. Now, autoimmune disease is the body at war with itself, right? Your immune system will recognize and kill your own tissues. For example, in type 1 diabetes, you'll recognize and kill your pancreatic beta cells, so they'll make insulin, so you have to inject insulin every day. So if you convince these cells to go where I want and do what I want when I want, you can then shift the balance from immunity to regulation, thanks to injecting these cells. And so how can I modify these cells to, you know, take them to where I want and do what I want once they get there? Like, what are the ideas to modify them? You know, I have the whole CRISPR arsenal in my disposal, genetic engineering, like, you know, sky's the limit. What can I do to these cells to make them into good drugs for this? Okay, I'll show you one example, which is, okay, who here has heard of CAR T cells? One person? Okay. So, CAR T cells are very famous in the cancer field because they have this chimeric antigen receptor, CAR for short. And what it does is it can now redesign, repurpose the T cell to go recognize and kill the cancer cell. So for example, this antigen C19, which I talked about before in B cells, so B cell derived leukemias have this receptor, but this receptor is not a traditional T cell target, it's not a peptide. So T cells could not see this. They have a very they have blindness in their eyes. But I can expand the horizon of what T cells can see using a CAR. And so the traditional example is you have a CAR T cell that will see and kill this cancer cell. Now, can I take this technology and use it not to kill, but to protect tissues? And so, if you can now imagine that you take this receptor and you design it differently, such that the signaling domain, which is the inside part of the receptor over here, this also helps make decisions. It's a signaling domain array. Luckily for us, signaling is like Legos. You can just keep assembling them and they kind of work almost independently. And so if you can find out by brute force almost, just like Yamanaki did many years ago, which signaling domain is best to keep our regulatory T cells suppressive and happy and surviving inside the body, we can now design these cells like little robots that wherever you have inflammation in your body, you can deploy them there and they will stop it. And so the focus Well, the work right now is in type 1 diabetes. So normally your pancreas makes insulin, controls your blood sugar. But if you're born with type 1 diabetes, you're born with rogue T cell clones that will recognize and destroy your pancreas. And so you have this fire going on. So can I design these extremely intelligent, extremely minute firefighters that will go and suppress these T cells specifically? Nothing else in your body, again. Specificity is the key to the immune system, specificity and potency. And so they'll suppress those T cells, and hopefully that will stop that inflammation, and now the pancreas will continue to produce insulin. So this depends on how advanced the disease is. So from my stem cell past, I guess, this will have to be a collaborative effort. If you just make stem cells into beta cells and put them into type 1 diabetics, you'll be like building a beautiful castle of sand in the beach, and the ocean comes and washes it away. But if you couple that with re-educating, rewarding the immune system, that sand castle can now retrieve the water, I guess, and the castle will stand for much longer. And so that's what we're working on right now, is to use these cells as living drugs to go wherever it is that you have an immune response, an unwanted immune response, organ transplant or immune disease or inflammatory disease, and then stop it from happening. And if it's a transplant, maybe it's an original organ, maybe it's an organ derived from universal stem cells. But either way, the idea is, again, to try to bring medicine to modify our body, that type of therapy. So not with small molecules, not with proteins, but with what we're made of, with cells themselves. So with that, I show you my most important slide, the thank you slide. So, thank you all for listening. I'll take questions. I'll start from this side. It's you.
You were talking about that, bringing in those T cells that you've modified so that they're immune to HIV. And you mentioned a clinical trial. What's that clinical trial about, creating the HIV resistance?
Yeah, so the clinical trial going in China where they modify T cells.
Sorry, go on.
Yeah, so the clinical trial is they modify T cells with CCR5 talons. So talons is a technology before CRISPR, but same idea. You just delete, you damage the CCR5 gene, then you put them back into patients because, again, these patients' CD4 T cell counts are so low that at the time, so knowledge about HSCs was not as vast then as it is now, or it was 2014, and so T cell is just the most obvious target, like as I said right away. If the T cell is sick, you're going to modify the T cell. That's like the immediate thing to do. So they did that, and this clinical trial is ongoing.
Okay, and do you have any idea on whether or not it's going successful, or any idea on like a guesstimate on how long this could take before this becomes a widespread successful thing?
Yeah, so I haven't released the data yet. So if you go on clinicaltrials.gov, you can follow it up. It's just like a rocket launch. So not really because I haven't released the data. But you can go and see. But so right now we don't know yet. But my guess is that these T cells are not immortal, unlike HSCs. And so HSCs are what we're pursuing. So some companies are doing this already with HSCs. So that'll be a more durable treatment. Both theoretically and in practice, because T cells will just die off and you have to do it again. But again, instead of taking 5 pills every day, if I tell you you just have to inject T cells every 6 months, that's a huge benefit. It might be very expensive, I should say. These CAR T cell therapies for cancer, how much do you think it costs? One single dose of CAR T cells targeting a B cell derived leukemia, like a bag of cells. How much?
125,000.
More.
Whoa.
220.
More.
600.
$500,000. $500,000 for one dose, which if it works is the only one that you need. So I should say that, again, this is going to be tough with I don't know anything about it, but with insurance companies, because you like to take insurance for the rest of your life, like pharma companies and insurance, but this one, so you're trying to do a one-shot thing. If you give you these edited HSCs, in theory they live forever, and then also to HIV, so that's the one-time, one-stop shop. Or in this case for the CAR T cells, once they eliminate the tumor, you should be cured forever. And actually, many tumors that used to be death sentences, they're now going to remission. And so it's a one-time thing. But however, it's $500,000. Actually, it's not even the most expensive therapy though. The most expensive therapy is $2.1 million. Yes. It's a therapy, it's actually based on RNA. So there's this disease that's called SMA, spinal muscular atrophy. So there's this gene that has this splicing problem. So if you fix that splicing problem, it costs you $2.1 million. So CAR T cells have a little ceiling to go. 20 years.
So from the way I understand it, the experiment in China kind of unleashed this technology, like without leaving much time for us to kind of debate like the ethics of how we were going about using it. So what steps do you think we're going to have to take, like, to ensure that this doesn't end up being used for things like kind of like designer babies, like
Yes.
changing eye color, like making your child stronger, things like that?
Okay, this is a very good question. So I think I should have started the timeline to show like how vertiginously fast it's built. Like in 5 years, I mean, I barely finished grad school, and CRISPR went from like a bacterial thing to making human babies. And so there's definitely not been enough time for ethical communities and scientists to understand what's going on and like the. So I think it's like a Pandora box that's been opened. And a technology like CRISPR-Cas9 that I was talking to perspective is not the first editing technology by far. Many others have succeeded like before it. But this one is so cheap, so easy to implement. Even if you don't do it carefully, a lot of off-target defects, lower efficiencies, but people just, it's like brute force it. And that's what happened right there. And so I think we have to be very careful. It will be a balance between make sure to respect the ethics and human rights. So maybe have some moratorium in the sense that if you're going to change eye color, maybe don't accept that. Maybe this kid was going to die from a terrible disease, maybe you want to accept that. But then there is a gray line, because even his own experiment, John Q.A., so he knocked out CCR5. You could imagine that, yes, it's a valuable gene. There's people walking around amongst us as CCR5 mutants and they're healthy. But obviously it has some impact, obviously. It's not like, there's no free lunch. And so actually, Nicholas just came out where if you compare CCR5 mutant people to CCR5 wild-type people, CCR5 mutant people statistically significantly live less long. We don't know why. So CCR5 is a chemokinase part of the immune system, so it must be important for the immune system. And so it seems that it's interacting like some viruses that, so for example, like West Nile virus is known to interact with a receptor. Like we're unlikely to get West Nile virus today, but maybe with climate change it will become a thing. And so imagine how terrible it would be, right? If you have a whole generation of people who are CCR5 knockouts, just because some crazy person thought, okay, let's just make people resistant to HIV, and then I don't think that 10, 20, 30 years from then, there's some new plague that wipes out all those probably extremely smart, educated, whatever people, but they were unlucky because of CCR5 mutation. And so these are very, very delicate decisions, because there's a reason why we have such diverse HLAs. It's not to be a pain in my neck. It's because if a plague comes, just by chance, at least a group of people will have just the right HLAs to present a viral peptide, really well, that will get eliminated. That's why such a diverse HLA, that's why so many HLA flavors. And this gene, same thing. If the gene exists, it's for a reason. It's just because some people in Northern Europe are to live happy lives, apparently slightly less long than other people, doesn't mean we should get rid of it just as a prophylactic. So I think that's like so germline versus adult. It's a discussion that needs to be had and is being had.
I was thinking about the mutation that you're doing in embryonic blood cell with CRISPR, and I was thinking about why you use other more trusted technologies, RNAi, or tolerance, I guess, would be more expensive. But RNA itself, the only difference would be it's not genetics, but it's still performing the same difference if you, it's still not the germline, and the embryonic CRISPR, and this was just after the production of the protein balance before. So if they're doing similar things, why not just use RNAi?
Because it's not permanent. So the appeal of this is that I do it once and I'm done, because it literally changed the genome. So every time this cell replicates, every time this other cell gets tries to enter a cell, that mutation is there. If you use RNAi, once it goes away, the RNA comes back up. Unless you want to knock in the whole RNA system with that specific one. So that's the thing, is that. And then so that's a good question in the sense that sometimes you can't just go with what works. For example, zinc fingers, the zinc finger nucleus, which came even before CRISPR, in some specific cases it worked really well. But there's a company called Sangamo that patented them in such a way that only they can use it, or otherwise it costs you millions of dollars. And so sometimes there's some non-scientific reasons why just one thing versus the next. But also the fact that it's just more versatile. Just because this one company screened 1,000 different nucleases to find the one that works, does not mean it's going to be feasible with CRISPR. With CRISPR, I screen 3 and I get one that works. And so you can just imagine, I think what Sidney Brenda said, as a famous scientist said, sometimes technology precedes scientific questions. So with new technology, you now can come up with new questions. And I think just the way we do biology now, it would be insane for me not to, recognize that the way we do biology has changed since CRISPR-Cas9. Because now, through my whole grad school, postdoc, many people, now knocking out a gene is no big deal. Making a mouse model is no big deal. Actually, even new model organisms, like sometimes I'm going to use mouse versus rat, because the mouse has this weird system worked out, but the rat doesn't. Now it doesn't matter anymore. Everybody starts from the same starting point. You take the rat cells, the mouse cells, the C. elegans cells, the human cells, and it seems to work everywhere. And so I think that's the main reason why CRISPR has got so many press, and I think deservedly so. It's just, the idea is just like it's this way. Everybody has access to it. Yeah, you got it.
Yeah, so like you said earlier, there's a lot of hype around CRISPR, but besides ethical problems that may surround it, what limitations do you see with CRISPR?
Well, so one limitation is off-target effects. So again, if you have different people, different genomes, there will be unknown off-target effect sequences that you don't know until you sequence it. So in my own experience, we try different guide RNAs to knock out CCR5, and I'm showing you the one that worked best. Maybe some people are not as careful with their work, and they'll just do the first one, and then you can go back to the old gene therapy days where people actually died because their genes of interest got integrated in the oncogene of tumor suppressor gene. So what if on top of cutting the gene that I showed you, it also cuts the tumor suppressor gene, like P53. So now, yes, you get the disease mutation corrected, but you also get a cancerous cell. And so off-target effect is very important. Actually, recently it's shown that on top of the small deletions, sometimes some guys also lead to large deletions, which were being ignored previously, but now are being taken into account. So I think just very carefully sequence your cells before and after, like a find the differences kind of thing, and make sure that only your sequence of features has been modified and nothing else. That's a big one. And I think delivery is another big one. So like in real estate, it's a location, location, location. In gene therapy, it's delivery, delivery, delivery. Like it's easy for us to take blood, grow the cells, modify them here, put them back in, but no one's going to give you a piece of their brain, or maybe you will, or of their liver or their kidney. So these cells, then we have to somehow inject the CRISPR-Cas9 in, and it has to go to the right place. So it is thing called AAV, adeno-associated viruses, and they have different tropisms, different preferences for cells. There are several studies that say, okay, this virus really likes liver cells. So you can think about designing a CRISPR-Cas9 package that is only delivered to liver cells. But it's been hard to reach. And so, again, I think we're a very exciting time because many of these things that used to be science fiction, they're now reality, or we know they will be reality in just a matter of time. So even if it's not true now, it will be true and possible in 10 years, 20, 50 years. And so I believe all these drawbacks can be addressed. You just need time.
How soon do you think it'll be common for parents to genetically protect their children from genetic disease?
Well, that's a good question. So I think it's already happening because, so that person in China said, like him now, so he went to the Human Genome Editing Forum in Hong Kong, and I tuned in, it was like watching the Super Bowl for science. So he was the first to say, look, I did this. And I was actually in prison at home. So what that tells me is that people have done it before him. You're just the naive enough one to tell everyone. And so I wouldn't be surprised if, rich parents will be doing this as we speak, and they just have, because again, maybe look into CRISPR-Cas9, so it'll be very hard to stop. And so I'll soon, I think it's today, and I'll soon in the open, I don't know, actually heard about this thing that came out in the news that they're trying to edit not germ cells, but sperm cells. Because like fertilized eggs, sperm cells I think have less ethical and law. I mean, the Harvard Law School, I'm sure consulted with them. And so if they can edit the sperm cell instead of the fertilized egg, I think it'll be easier and less ethical problems. But the end result is the same, right? So sperm cell is going to fuse with the oocyte and give you an egg. And so it's kind of the same thing. It's still making a human being in the end. And so again, I think it's just I wish I had better answers for your questions, but it's really a Pandora box that has been opened. And all we can do is be very responsible, have this talk as much as, so that's why, everything I showed is unfiltered. I'm showing what I'm doing as of now, what other people are doing as of now, because the more we hide things, the more illegal or not so ethical things happen. So these debates have to be happening in the open, number one. And number two, by educated people. You cannot form an opinion if you don't know what's going on. So I think you have to be as open as possible, say, I'm doing this is what it could mean, what do you think? But what you think should be the third step, not the 0th step. which is what happens sometimes. Any more questions? I don't think so. Yeah.